BMS enables you to register and manage batch data, and it can be used to build a prediction model from experimental data and optimize process conditions and parameters. In this session, we'll learn how to register batch data based on existing Labnote. Let's click Register button. Select Project at Labnote first to register a Batch Labnote. The new Batch Labnote will be subordinated to the selected project at Labnote. Register window is composed of two sections. Labnote Components section provides the standard conditions and input fields for Labnote registration. Available parameters depend on the selected project. Right side window allows you entering general information. It contains information, category, reference, and note. Information section consists of Labnote name, document number, status, and keyword. Enter Labnote title and document number is not prerequisite. Initial status is preliminary. Enter keywords carefully because you can search the Labnote by keywords in the search menu. Category is an index to classify the Labnote, and also you can search Labnote by category information. You can add a category from Labnote Component section to Category panel. Expand section category folder and add initial development category by drag and drop or double-clicking. Next, register reference information related to the Labnote. According to reference type, select paper or patent, and so on. The supported reference formats are file, text, and website URL. At the Note section, you can add unformatted information such as text, table, or images. Once all data has been registered, click Save button. You can import batch experiment data into datasheet. Multi-flexible datasheet is provided. The datasheet will be registered as new or based on Labnote. Labnote-based sheet creates datasheet based on material, precaution, method, observation variables from Labnote. New sheets generate new datasheet based on Excel file. At first, click Labnote-based sheet button. Select variables to add to datasheet. The variables are sub-items registered in the workflow stage. Select items and click Generate button. The datasheet is created with the selected variables as column header. New column will be added into left side of the selected cell. Click on the right mouse button in the upper left corner and then click Add Column button. Insert the column name to experimental name and select the data type to text and click OK button. To save the batch data, click Save button. Using Export button, download Excel file and fill out the experiment result in the specified format. Copy the data except for column header and then paste the data in the first column of the data sheet using Ctrl-V. Click Save button again. Click on the right mouse button in the Datasheet tab. Click Rename button to change the name of datasheet. Next, I will show you how to generate new datasheet based on Excel file. Move to Information tab. Click New Sheet button. Add Sheet dialog opens. Open your Excel file and copy the data, and then paste into the Add Sheet dialog using Ctrl-V button. Select column header using mouse drag and click the Pick Column Header button. Click Next Page button. To map variables to material, method, and observation class, select variables and click the right arrow button to move to each class panel. For example, lithium cobalt oxide, 1-methyl-2-pyridine, polyvinylidene chloride, and cathode active material slurry map to materials. Agitator, pressure, and aging C rate map to method. Lastly, energy density and power density map to observation. The remaining experiment name is not part of any data class. Click Next Page button. Select data type for each column. Data type means type of value entered in the column. Category type is assigned by administrator. Assign experiment name to text and pressure to level. When you click Generate button, the Excel file is imported. You can add date or file type data. After click on the right mouse button in the upper left hand corner, click Add Column button. Enter the column name to Ragon plot and select the data type to File and click the OK button. Click plus button in the Ragon plot column and attach an image file. If you click Preview icon, you can see the attached image. Click on the right mouse button in the sheet name and click Rename button to change the name of sheet. To save the data, click Save button. By now, we have learned how to register Batch Labnote and datasheet. Thank you.